and one willing to lo-fi, so this review isn't too boring. Okay, so let's start with kind of the first thing that you want to be talking about. I guess one of the engagements I can do for you is when we're talking about, something like gel electrophoresis, and also with the PCR, but mostly just gel electrophoresis. The main thing you want to know about gel electrophoresis is that the way that we use this in real life is when we're talking about criminology, right? That's one of the most common methodologies for using gel electrophoresis. So it helps scientists pretty much identify different people, okay? And in the more criminology way, it helps them identify a criminal, right? From drops of blood, and then you would input that into the gel electrophoresis technique, and then you would get a certain output. Now, what I really want you to do is, figure out how they're able to differentiate between different people, right? And I mean, the best thing I can kind of give to you is your image from the slideshow you gave me, right? So we're looking for, this slide says, who did it, right? I mean, it's circled male 1. Okay, so like really why can't I? So really your clue here is, looking at who matches a specific DNA sequence that you're looking for, right? And it just helps you narrow down and isolate specific types of DNA, right? That's the point of gel electrophoresis. Okay, what it really does is it isolates DNA in the sense of separating those DNA fragments so everything is very easily analyzable, if that makes any sense. Okay, so that's enough talk about this. Let me delete all this. Soft eraser just doesn't do anything. Where's my select tool? I'll just leave everything like this. Oops, I'm saving. Oops, I'm saving. Okay, so first thing that we're going to talk about is DNA manipulation. Okay, so on your slide, the big thing that I saw was the restriction enzyme. Okay, and when we're talking about restriction enzymes, it might help you to think about it in terms of, I call them molecular scissors. Okay, an application of restriction enzymes is, I'm not sure if you've heard of this before, but with CRISPR gene modification, we use restriction enzymes to kind of cut into that DNA and cut it at a specific sequence, right? So think about this holistically, right? We're saying a restriction enzyme, so it's going to be, restricted to a certain sequence, right? It's just going to be restricted pretty much to a certain or a specific sequence of DNA, right? And then when you think about it, right, if you cut that DNA at a specific sequence, there's going to be two parts of that DNA, right? If you cut a DNA, obviously now you have two sides you're working with, right? And one of those sides is the sticky side, okay? We call that a sticky side to the DNA. And that's what other DNA can recombine to, okay? You have your different types of DNA, or, different endings of DNA. What is it? Uracil. It's a diacyl, I don't remember the names, but you know your A, C, T, and G, right? Those are your recognition sites. And when you're trying to create, you know, complementary strands of DNA, you want to match those to the correct recognition site. So, you know, you match A to T and C to G on DNA, right? And those recognition sites, they bind to the sticky end, okay? So I'm gonna just draw a really bad example. Okay, so say you have your DNA, right? You have your DNA structure like this. It's not the DNA. This is kind of like what the DNA is attached to. And my DNA molecule is gonna be in between these lines, okay? Like a ladder. Think of DNA like a ladder. And so we're thinking about DNA like a ladder, and then some of these are going to be binded from the right side, and some of these are going to be binded from the left side, right? And then obviously we have the other kind of ladder for the DNA, and I'll just match everything like this. So we have, you know, these different DNA, and I'll say this is C to G, this is also C to G, this is T to A to T. Well, if it's A to T, then I'll have to match it the other way. So because I'm going to one of them going to the left side, one of them going to the right side, and then we have G to C and then G to C. Right, so we have matching strands of DNA, right? And so let's say we want to cut it. Let's do this in red. Let's say we want to cut your DNA. Why isn't it red? Hello? What the heck? Okay, whatever. So let's say you want to cut your DNA, right? So let's say I'm going to cut it like on this one. So I'm cutting my DNA from this strand, okay? And so if I cut my DNA from that strand, right? Where is my DNA? What is the only strand that's going to be left? And it's going to be this top strand, right? This top strand here is the only one that's going to kind of be left over. So when I redraw this, not going to draw the whole thing, right? But it's really we have all of this stuff here, right? And then we have only one of those DNA fragments still left over, right? Our C and G DNA fragment. And the rest, because we cut it at that joint, we're only going to have the ones from the right side. Right side, sorry, left side. All of these ones. These are the only ones that we have left, right? And I'm gonna match them to the ones we had before. So C, C, T. I forgot to add one more, but it's whatever. You get the point here, right? So we cut it at this joint, or that DNA intersection, and then we have this other kind of, you know, this other part of the DNA on this side, right? So we're gonna have G in that, G there, A, T, and C, right? I'm not gonna draw everything, right? And then one of these parts are going to be the sticky end, right? So we have some sticky ends of Dna, and it helps, you know, to recombine them onto the complementary strand of DNA, right? So that's just kind of the sticky side, or the sticky end of the DNA. You also have, you know, we have blunt end, right? But we don't really want blunt ends, okay? Because if you have blunt ends, it's harder for the DNA to recombine, right? And obviously DNA recombination is one of the most important things for biology, or for just molecular biology in general, right? If we cut a piece of DNA, we typically want to, you know, recombine other different DNAs, you know, to create something new, or maybe to just recreate something, right? DNA is all about manipulation. So it's important that we want to get sticky ends rather than blunt ends, okay? And that's why it's important that we have restriction enzymes, because it helps us cut that DNA once again at a very specific site, right? If we didn't have restriction enzymes, there would be no way to tell that enzyme to cut at a certain sequence. We just wouldn't, there would be no way. So that's why this is one of the most important things in molecular biology, right? The restriction enzyme just helps you cut at a specific point on that DNA sequence. Okay? Let me make a, what the heck just happened? I don't know what's going on. I never used software before. It's a new one. I'm gonna make a new. What? Okay, it's whatever. So let's make a new page. Oh my god. There we go. There we go. Okay. So I'm in a new page, and now let's talk about gel electrophoresis. Okay, and this one's gonna be hard to draw, so I have a picture pulled up already. So that's what gel electrophoresis looks like. Okay. And so let's talk about this. What you need to know for gel electrophoresis is that DNA by itself is negatively charged. Okay, DNA has a very small negative charge. And what do you know about charges in general? How do they like to get attracted to other things? Well, DNA wants to move to the positive side, okay? And we call the negative side the cathode. If you're confused about negative, it should be different actually. I think the anode is, that's the anion, I'm sorry. Well, then it doesn't work. But think about this, you know, cats, they scream a lot, so you attribute them with the negative part, and then the anode, I guess you just gotta remember. It's weird because the anion is actually the negative charge. I guess that differs for anodes and cathodes. Okay, but we know that DNA is negatively charged, right? So it wants to go to the positive part, and that's the way that gel electrophoresis works. Okay, we isolate DNA by using a centrifuge. Okay, you'll probably do that in a bio lab sometime in college. So we isolate DNA through a centrifuge and we put it into gel electrophoresis. Okay, we literally inject DNA into a buffer, right? Usually of water and something else, right? Or water and gel and something else, and it helps to move that DNA to the other side. Okay, and the speed at which this happens differs, right? The pathway for the DNA is set. It's a set path, right? And so if your DNA is large, obviously it's going to take a slower time, right? But if your DNA is smaller, it's going to take a faster time. It's going to be faster to reaching the other side of the gel electrophoresis machine, right? It's going to take faster, or takes a slower amount of time for a smaller DNA fragment to reach the positive side, right? And you'll actually see that denoted on the electrophoresis. Smaller fragments, they move farther. Okay, so if I have a gel electrophoresis, for example, and this is my negative side, and there's these little cells, or my wells, on the negative side, and I inject DNA into all these wells, right? Well, some of my DNA might do something like this. Okay, it might stop here. My smaller DNA fragments might they might even finish and go all the way to the end, right? Some of them might end here, might end here, and some of them, you know, they could, break in the middle. Some of them can do that, okay? And we indicate that differently when you're actually viewing the gel electrophoresis. I don't think that'll be tested though. That's more of a lab thing, not really important for practical use. But in general, what you want to know about gel electrophoresis is that you inject isolated DNA into a gel buffer, and then because it's a negatively charged DNA fragment, right? DNA is always negatively charged. It gets pulled towards the positive side, right? And what does that mean? Well, that means there has to be a current somewhere, right? So that buffer typically has an electric current, or I'm not sure the buffer has it, but there's an electric current somewhere in that electrophoresis that is pulling that negatively charged DNA onto the positively charged anode on the opposite side. Okay? So just to ask yourself this question, what would move farther? Smaller DNA fragments or larger DNA fragments? Right, so I'm not going to tell you the answer. You can go back into this if you want to really know, but just know that. What moves farther, right? Larger or smaller DNA fragments? So third part of this, I'm not going to read the page as well again. I'm going to use this software. Cancel. So next thing we'll talk about is the PCR. Okay, this is one of the biggest things of bio as well. Okay, so now we're talking about the PCR, or the polymerase chain reaction. Okay, you just call it the PCR because that takes way too long to say. And the point of the PCR is to copy DNA. Okay, so if restriction enzymes were used to manipulate and combine DNA, and gel electrophoresis is really just used for a DNA analysis, right? This one is used to copy DNA, okay? So this is what we're going to use to literally copy DNA. And this is important when we're trying to do DNA fingerprinting later. So what is the PCR, right? What does this consist of? Well, the PCR is a 3-step process, and for high school biology, you're not going to have to know the specifics, okay? Specifically, denaturation, there's multiple steps to this. The first one happens at 95 degrees Celsius, and then yada, 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 right? But you don't need to know that kind of stuff, to my knowledge. I didn't see it on the PowerPoint notes. Well, this first step is denaturation. Okay. Second step is the annealing. And the third step is extension. Okay. And I think these are pretty self-explanatory. But I'm sure you've heard of heat denaturation. And heat denaturation just means when heat kind of just separates proteins, pretty much. Not exactly the most accurate definition, but that's pretty much what heat denaturation is. And it's the same thing with DNA, right? So with denaturation, when DNA is put under significant heat stress, or particularly 95 degrees Celsius, that heat is going to separate the strands. Okay? So it's going to separate those strands of DNA. And that literally looks like, if we have a DNA molecule, okay, I'm going to simplify it, and I'll just make it like a ladder. Right? With denaturation, this literally becomes something like this. Okay? So the DNA literally gets separated. Okay? And the second part of denaturation is annealing. So I'm going to kind of delete this now. Excuse me. There we go. So the second part is annealing, and this is when primers, okay, so DNA primers are going to attach. Now you might be asking, what is a DNA primer? Okay. Well, a DNA primer is kind of like, I want to call it the building block of DNA. Okay? So primers, kind of like their name says, they kind of prime the DNA polymerase to start building DNA. Okay? And so that primer, it's going to bind to, so when we have that denatured DNA, right? We have a denatured DNA molecule. That primer is literally going to attach. Okay, it's going to attach to the denatured DNA. Okay, and it's only going to attach to certain DNA. It's not going to attach to all of them, right? So what that looks like is you might have a DNA thing that goes like, whoop, right? And then you have the one on the bottom that goes like, whoop. Oops. Right? So, oops, this is actually the wrong step. I'm sorry. You have things that go like this. I'm sorry, I skipped a step. Okay? So you have these primers that bind to the DNA molecule. Obviously, your DNA molecule is going to be a lot larger than this, right? So usually, what, like 20 something of strands that are on there. But they're going to attach to certain parts of the DNA, and they just keep attaching, they keep binding to these denatured strands of DNA. Okay, so they just keep attaching onto those. And actually, I should probably pull up a picture of this so it's actually better to see. Let's see. There we go. Probably better this way. So I'll just put on them for the screen for a little bit. We have denaturation when the DNA molecule gets denatured by heat, and then we have binders, or primers that bind to those denatured strands of DNA, right? And then what do you think polymerase extension means? Right? We're literally just expanding or extending the DNA molecule from polymerase, okay? So the reason why, again, why the primer is important is because it primes the DNA for DNA extension, okay? Without annealing, extension won't happen. So you need the annealing step to happen for extension to happen. You need denaturation for annealing to happen, right? We cannot just attach primers without the denatured DNA, right? It's a very, very picky process. Okay, so all these steps are very important for the next one to proceed. Right, so we're thinking about these primers, right? In DNA extension, we're going to just extend these primers outwards, right? And it's going to fill up that DNA molecule. And you saw in that picture there, right? I'll pull up the picture again. It's going to fill up the entire DNA molecule. Now you'll notice that it's missing two of them on the left and the right. Well, that's what helps the DNA bind together, right? So we don't need... How will it be able to bind the DNA if everything has a primer on it? Right? Just can't, right? So there needs to be, you know, two little strands of DNA that can kind of bind together. Hear that? Okay, perfect. And again, with extension, oh my god, the pen is gone. With extension, this is just our DNA polymerase that builds new DNA. Excuse me. Okay, so first we have denaturation, where the heat denatures the DNA. It literally separates, I would like to say separates the DNA into two different parts. Then we have annealing, in which primers attach to those separated strands of DNA. And an extension, in which DNA polymerase, because of those primers, it can extend those primers out. Okay? It extends those primers out, and in that sense, that builds more DNA. Okay, so we're building more DNA because of the primer attaching to the DNA strand. Now, why is that important? In this particular mechanism, DNA doubles. Okay, this is how we double DNA. Once again, what is this used for? DNA cloning, right? So this is how we clone DNA. And if we clone DNA, what do we get? We get two of the same thing, right? So PCR is important because it clones DNA. You don't really need to know, I believe, like what a primer does and all that stuff. I'm just telling you for the sake of, you know, just telling you, right? But you don't need to know actually like what they are. I believe so. If you do, then maybe you should. But that's pretty much it for the PCR. Now, see again, primers are needed because DNA polymerase needs the primers in order to start building DNA. Right? So when you denature DNA, that strand no longer exists, right? We literally denatured them. And so primers kind of, not reattach, but they let DNA polymerase, you know, kind of add on to that strand now. right? And it cannot add onto that strand, specifically cannot add the nucleotides onto that strand if it has already been denatured, right? It needs something, it needs that binder or the primer to attach those nucleotides to. Okay. Okay. So that's done now. Let's move that page. Oops. Oops. And if you have any questions, just let me know. If I explain anything that when it's not very clear. So, to my knowledge, the next part talks about DNA fingerprinting. Okay, so we have DNA fingerprinting. So, this is pretty easy. I don't think I need to go too in depth in this, but this is just in your slides, for the most part. Just tells you that, you know, we're going to use unique DNA patterns, okay? And to get unique DNA patterns, you know, and we're going to need the PCR and gel electrophoresis, right? Most importantly, we really do need gel electrophoresis, right? Because something like this, right? We can clearly see the differences in DNA, right? Like if we want to match, you know, what are we looking for, right? And suspect 2 from his electrophoresis matches the DNA crime scene sample, right? So that's why it's important that we need to know the PCR and the DNA polymerase mechanism, because in real life, it's used for fingerprinting, right? This is what they use actually when they fingerprint you for jobs, right? If you work at a church, they fingerprint you and they go through the PCR and they go through gel electrophoresis to get your fingerprint scanned. Okay, so nothing too much to talk about there. I can just delete all this stuff. Next part is talking about recombinant DNA. Pretty cool stuff here, actually. So, what do you think recombinant DNA sounds like? We're recombining DNA, right? So, recombinant DNA is DNA from all these other, different organisms combined, right? I can have, you know, DNA from, you know, a sheep plus a bat. I'm just bringing up examples plus a cow, okay? We can have all these different DNA from different organisms, and we can combine them, okay? Now, you can't really use, this is a bad example because you don't really use this kind of DNA. You use bacteria DNA, particularly. You don't really use a cow DNA. I just brought up, I don't know why I brought that up. But think about it as you're just using DNA from different organisms, right? So we're going to combine different DNA, usually from bacteria, and more particularly, it's from the plasmids in bacteria, right? We're going to, use a DNA plasmid from a bacteria. Say, for example, we're using E. coli, right? The plasmid, we're going to literally remove that plasmid. And how do you think we remove that plasmid? It's a selective cutting. You learn something to do with selective cutting, right? And that is the restriction enzyme, right? Most things to do, right? If you're cutting something out of DNA, it's because of that restriction enzyme, right? Very, very important. This is the basis of all DNA modification. It is that restriction enzyme. So let me draw this out for you real quick. Let's say we have an E. coli molecule, right? Let's say this circle is our E. coli molecule and this outer layer is the plasmid, right? We're literally going to remove this plasmid. Okay, we're going to remove this plasmid. And on the right side, we have a strand of DNA. Okay, so it's like a strand of DNA on the right side. And we're going to cut it into different pieces. Okay? We're going to cut it into different pieces with what? Cutting again, that's the restriction enzyme. Right? So I'm going to cut it into a piece that we can manipulate. Right? I cannot manipulate like this whole thing. It needs to be, cut down to a smaller scale. And we do this so we can isolate certain genes, right? And let's say, for instance, we're trying to isolate the insulin gene in humans, right? So I'm going to splice that human insulin gene with the plasmid from the bacteria, okay? And what do we create here? Well, we create this sort of hybrid DNA in which this portion right here, all of this, I can do this better. Let me use the fill tool. Change this to red. So all of this stuff, oh my god. Try again. All this stuff in here is, finally got it. All that stuff in there is our, we call that the recombinant DNA, right? So this whole thing is the recombinant DNA. This particular part, this red part, that is the human insulin gene, right? We're literally splicing them together. And what is the significance of this though? Like, okay, that's cool, right? That's cool, but what does that do? Think about this now, right? We're going to reinsert this plasmid into the same E. coli cell. Okay, because what do we know about bacteria? They replicate very quickly, right? Bacteria, they replicate super, super quickly. That's why they're very hard to get rid of in our body, right? So these bacteria, they're going to replicate quickly, and they're going to replicate, think about this, right? We're going to insert this back into an E. coli bacteria, or E. coli cell, right? So we're inserting it into that E. coli cell, and then that E. coli cell with this new, remember this is called the recombinant DNA hybrid. Plasmid hybrid. Right? We're putting that into the E. coli cell, and then that E. coli cell, that bacteria, is going to replicate with this human insulin gene. We're literally duplicating human insulin genes by using a plasmid from a bacteria, right? So that makes a lot of sense, right? So we're just cutting out the plasmid, and we're cutting out, or we're isolating the human insulin gene, all from using restriction enzymes, right? So we're going to splice them together with DNA technology, right? And then we combine that to make a recombinant DNA structure, right? With the human insulin gene, right? That red part is the human insulin gene. And we're going to insert that back into an E. coli cell so that E. coli cell replicates over and over and over again with the human insulin gene, right? So now we're creating clone cells that are capable of producing human insulin. And we know that insulin is important because, you know, we have different conditions that require insulin shots, right? Diabetes. Right? Why is it important for diabetes? Because we develop something called insulin resistance, right? Where our bodies naturally just start creating less insulin. We have insulin resistance, right? So we need more insulin in our bodies to regulate our blood sugar. So it's important that we have insulin, and that's how. This is one method of doing it, right? We can do the same thing with pig insulin. Same concept, right? We don't need to use human insulin DNA. We can use pig insulin. They're very biologically similar to us. Okay, well that's the thing with recombinant DNA. One last thing with this is that it's not limited to just insulin, right? Think about it. It can also create different hormones, right? We have different hormones in our body, right? All those hormones, we can also just use a plasmid from bacteria and combine those two together to create that recombinant DNA molecule, right? And we create that recombinant DNA molecule, and we can produce bacteria that replicate hormones too, right? So it's not just limited to insulin, it also extends to hormones. Okay, so that's that about recombinant DNA. That's ugly. Okay, next part is cloning. I don't think cloning is that too complicated. Cloning, obviously, right? We're just creating an identical genetic copy, right? So we're talking about cloning. It is an identical genetic copy. Identical is the key word here. Now, transgenic, okay, this is pretty cool. Transgenic means it's from DNA or genes from another species. Okay? When you're talking about things like genetically modified organisms, we're usually talking about transgenic modification. Okay, we're inserting foreign DNA into a DNA sequence, right? So we have foreign DNA being inserted into a parent DNA. And that's kind of invasive. That is invasive manipulation. CRISPR does something different. It's just like a little tangent. CRISPR uses non-transgenic DNA modification. So it uses DNA literally from the same sequence into that same sequence. If that makes any sense, right? We have DNA from the same organism being modified from the same organism. We're not using foreign DNA, right? So that's the difference between those two. Transgenic means we're using foreign DNA and inserting it into a DNA sequence. So that's pretty much it with cloning. If you want to look at some diagrams, you have your notes. Now, what are some applications of this? I guess think about organisms that are not natural in this world, okay? Glowfish, those are not natural. Okay? Crops that are naturally pest resistant, those are not natural as well, right? And there's no way for those to exist without having transgenic modification, right? We need to insert a gene from another species that has some form of, in glowfish, right? A gene from something else that has some form of bioluminescence. And with pest-resistant crops, we need a gene from some other, maybe some other animal or some other plant that has natural built-in pest-resisting mechanisms, right? So that's what transgenic modification is for. To create really desirable and exact modifications. So the last thing is, another thing was, I think was gene therapy. To my knowledge, was gene therapy. This is not very complicated either. So we're talking about gene therapy and also applications of in biotechnology and also agriculturally is what I saw, right? We had some agricultural applications as well. So think about, you know, gene therapy. What does gene therapy sound like to you? Therapy for genes? I mean, pretty much, yeah, that's what it is. It's therapy for genes. We're trying to fix something, right? You go to therapy to fix something. So what is gene therapy? We're fixing defective genes. Okay? So your gene might be defective, right? And it would undergo gene therapy in order to fix that gene, right? I don't think it needs on the mechanism, but that's pretty much what it does. It's just a way that we fix defective genes. And I'm sure you've heard of things like sickle cell anemia or cystic fibrosis. These are all conditions that are caused by gene malformations, right, or defective genes. So we use gene therapy solutions or gene therapy technologies to kind of help or assist with these conditions. And with biotech, think about medicine-wise cancer treatment, right? A defective gene is also like a defective cell, right? Rapid growth of a cell, that's considered a defective gene, right? There's a gene in there that went haywire and caused that cell to grow at an abnormal rate. So in biotech, gene therapy is used to treat cancers, right? Gene therapy is also used to treat insulin resistance, right? We just talked about the form of using the recombinant DNA, right? We use that recombinant DNA for you to use in biotech, right? If you need insulin, we can do that, right? we can use a recombinant DNA to create insulin for you. Now in agriculture, think about all the kind of foods you eat, right? Pesticides, well, not pesticides, but if you want to create something that, or a crop that has higher yield, it has to undergo gene therapy, right? In order to produce higher yield, you need to modify that a certain gene, but that gene in that instance won't be defective, right? We're just trying to correct it into what we want, right? So we might modify that gene to create more yield. It works the same with resistance. You want to resist something in the weather, we would modify that gene to be more correct, or modify that gene in a way that helps it resist certain weather conditions or certain pest conditions, right? That's pretty much all I have for you. I'm not sure if that was too helpful. It's kind of hard to teach without seeing questions. But that's pretty much what I know. Right, so as a review, the main topics for today was restriction enzymes, PCR, and gel electrophoresis. Right, restriction enzyme, think about that as what? Your molecular scissors, right? It's selective cutting on a DNA sequence, so we can modify it for later. What's the PCR used for? PCR is a way to copy DNA, right? We're just copying DNA, and it has 3 steps. Right? Make sure you know those 3 steps: denaturation, annealing, and then extension. Right? Now, the third thing to talk about, main point for today, was gel electrophoresis. Right? Think about that like DNA kind of racing to the other end. Right? DNA is negatively charged. It wants to go to the positively charged side. So we put that in a well, in a gel electrophoresis machine, and then an electrical current pulls that DNA onto the anode, right? And then based on the length of the DNA, we can match that DNA with other documented DNAs, typically used in crime analysis. So yeah.